Moving right along, our next session is the Peter Como Platform Symposium. The symposium, which began in 2007, provides an opportunity for researchers to present their valuable findings in clinical HD research to event attendees through poster sessions and platform presentations. HSG named the symposium after Dr. Como, who played a key role in the founding of both the HSG and the symposium. I'm happy to welcome Dr. Aaron Verstemming from UT Health in Houston to introduce our speakers for this session. Dr. Verstemming.
Okay, thank you. Thank you for the beautiful tribute to Dr. Schulson. That was incredible. And to our most recent speakers, that was just riveting, really exciting. So I would like to first introduce our amazing Publications Committee. I'm honored to serve as the co-chair of the HSG Publications Committee, along with my friend and colleague, Dr. Clement Loy, far, far away in Australia. Our wonderful HSG Publication Committee members include Amy Brown, Eric Molo, Paolo Moretti, Kathleen Shannon, and Sandra Skinner. We sincerely appreciate their assistance in selecting our poster and platform presentations. We had 95 abstract submissions this year. Thank you to everyone that submitted your work. As has been mentioned, the Peter Como Research Symposium provides an opportunity for clinicians and researchers to share their valuable findings and exciting work with our HD community. The symposium was named after the late Peter Como. Dr. Como was a renowned neuropsychologist who made an indelible mark on HD-related care and research. We are incredibly grateful for his contributions to the HD community. So we have two exceptional speakers today. They're going to share their work. Our first presenter is Dr. Molly Zhang. She is a faculty member in the Division of Neurobiology, the Department of Psychiatry and Behavioral Sciences at Johns Hopkins University School of Medicine. Her research centers on understanding the pathogenesis and neurodegenerative diseases and developing biomarkers and therapeutics with a primary focus on HD. With extensive expertise in translational neurobiology, Dr. Zhang is actively engaged in HD drug discovery, including hit screening, lead optimization, and target validation. She has identified and validated novel therapeutic targets, making significant contributions to the HD research field. Welcome to Dr. Zhang, who's hopefully amazing. Okay, love it.
Thank you for the nice introduction. So for this talk, the original speaker, Dr. Li Shanling, cannot attend this meeting. Thus, I will present on behalf of Li Shan from Dr. Zhong Pei's lab. This is a first-in-human clinical study about a ANOVA strategy called ER 2001. It's a self-assembled Huntington siRNA. Also, it is an exosome-based therapy in earlier manifest HD patient. No disclosure from myself. As we all know, siRNA is a powerful tool to inhibit the interest target. However, there are barriers for siRNA drugs, mainly from the cross from the plasma membrane and the release of the siRNA from the endosome. So in the right panel, here I show the current siRNA clinical trials. You can see after extensive development, currently the most successful organ for delivery is the liver, except beyond the liver, there still is a big challenge. So to develop the siRNA efficient delivery system. So some observations from Dr. Chen's group, they found that actually microRNA can stably expressed in the actual cellular fluid through the exosome. Also, the microRNAs can be transferred between cells via the exosome. Those evidence suggest the exosome might be a delivery system for the siRNA. So compared to the virus and the nanoparticles, the exosomes has more benefit to direct deliver to the cytosol and with less toxic and immunoresponse. So the current conventional exosome siRNA assembled strategy is culture the cells, express the interest molecule, and then you collect the medium, extract the exosome from the medium. For this strategy, is pre-assembled in the vitro. That's why it's not physiological. Also with high cost and labor intensive. To develop the in vivo self-assembled siRNA exosome delivery strategy, so Dr. Chen's group, they designed a construct. Here you can see with 3 components. The first component is a promoter. So they pick the CMV as a promoter here because CMV is a dual driver, can drive the protein and the siRNA same time to express in the mammalian system. The second part is the RVG tagged LAMP2B. So the RVG is a ribose virus glycoprotein. So this tag can bind to the nicotinic alpha-7 receptor to target neurons and glias. The LAMP2B is an exosome membrane protein. Then the third part is the Huntingtin siRNA backbone. So this whole plasmid, if you inject to the blood, it can accumulate in liver and reprogram liver to produce the exosomes contains the Huntingtin siRNA and release to the blood circulation. Since the exosome is small and also is biogenic, that's why it can pass the blood-brain barrier, get into the brain. Then once the exosome into the brain, the RVG tag will guide the exosome to the neurons and glias and release the siRNA to target the messenger Huntingtin. So this strategy, the advantage is minimal invasive if compared to lumbar puncture and the brain injection. Also, it's because it's bio-generated. That's why it's more less toxic and less the immunoresponse. Definitely, it's also because it's produced by the subject itself. Also, the physiological concentration with that range. Also, this has a high on-target activity and low off-target effect. So to prove if this construct, this system, if engaged to the target, so the Dr. Pace group, they inject, IV inject the construct to the bank HD mouse through the tail, and they did 6 injections once per week. And then they observed the mutant Huntingtin levels at the indicated time point. You can see here, in most time point, in both cortex and stratum, the mutant Huntingtin reduced about 50%. Next, they also did the safety study on monkey and the rats, so also showing the safety of the ER 2004. Encouraged by those preclinical study results, next, Dr. Pei's clinical group, they did an open label dose escalation earlier phase 1A study of the ER 2001 in the HD early manifest patients. So the primary outcomes, so they observed the safety, all the safety measurement. And for the secondary outcome are the PK and the PD studies. And the endpoints include the functional study and the biomarkers. So for the dosage, they have a strategy called escalation. So they first tried the lowest dosage on one subject. Then if the patient will tolerate the dosage, then they increase the dosage for the other patients. So it's up to 0.32 milligram per kilo. For this 1A study, they first they initiate one injection, waited for 2 weeks. Then they did the once per week, and totally 6 weeks, and then wait another 2 weeks and did the last injection. That's why totally 8 injections. So totally they recruit 10 HD patients. So for the primary outcome, the safety, you can see here, there are totally 62 the AE events they observed. And most of them are grade 1, few are grade 2. That's the ER 2001, the toxicity is acceptable for future clinical trial. For the PK results here, you can see after the injection in plasma, either the DNA and the siRNA, their peak is at injection about 1 hour. Then the DNA decayed quickly, and the siRNA can maintain a relatively high concentration up to 12 hours. Since there is no available mutant Huntingtin CSF assay in China, so Dr. Pei's group, they use their 293 stable cell line with mutant Huntingtin expression with a flag tag. And they treated the cells with the exosomes from the mock control and the healthy control, and before treatment and after treatment. You can see the treatment can decrease the Huntingtin, mutant Huntingtin about 40%, and also dramatically decrease the Huntingtin aggregates. Then the endpoints functional study, you can see here, there are motor improvement across the 3 dosages except the lowest dose. The cognitive outcome also suggests the protection from this treatment. So currently, in China site, so the phase 1B started the recruitment. So for this 1B study will be about 30 patients, and they will use 3 dosage from the 1A results. For the US site, now currently is opening for partnership to support this study. So thanks for the whole team, the excellent work for this study, and I will stop here for suggestions and questions. Thank you.
Sorry, we're OOT, out of time, so you'll have to save your questions for later. But that was beautiful. Thank you so much, and Fantastic presentation. Okay, so our next platform presenter is Brian Beers. Brian is an Associate Vice President with PTC Therapeutics and the Global Program Lead for PTC 518 program. Brian is a 24-year veteran of the biotech research community. He's been involved in the design, implementation, and management of clinical trials in a variety of therapeutic areas and several orphan indications. Fun facts, Brian graduated from the University of Illinois with a bachelor's in microbiology and chemistry, and he's an avid traveler, hiker, and golfer. He currently resides with his wife and 2 children in the one and only Nash Vegas, Tennessee. So welcome, Brian. We're excited to hear about the pivot data.
Thank you, Dr. Verstemming, for that introduction, and thank you to the HSG for allowing me this opportunity to present to you our interim data here today. All right. Okay, so my presentation today here is on our 12-month interim results that we have from our Pivot HD trial for Huntington's disease. Just my disclosures really quickly. I'm obviously an employee of PTC and on stock. Also, in all of our talks that we've had here recently, we talk about PTC 518, but an exciting sort of milestone for our program is that our INN naming is actually complete, and we are now referring to PTC 518 as Votoplam. So Votoplam is what I'll say moving forward, but don't penalize me if I accidentally go backwards to PTC 518. This is the first time I've actually given a talk where I get to say Votoplam. So just a couple of slides here to level set with everyone here who's in the room who hasn't heard about our program here in the past. What we are here is a program with Votoplam. We have an oral splicing modifier. So what Votoplam does is it splices Huntingtin mRNA, which leads to subsequent lowering of Huntingtin protein. And we've shown this in animal models, in prior conferences. We've shown this in our phase 1 data, a really nice dose-dependent lowering of Huntingtin mRNA in healthy subjects, which is really what brings me to the talk here today, which is the phase 2 program. So a brief description of the phase 2 study design. So the study's design is actually, it's a 4-arm study, but what I'm showing you here too is just, here today is the 3 arms. We have 1 dose at 5 milligrams, 1 dose at 10 milligrams versus placebo. And ultimately, Pivot HD is a year-long trial. It's broken out here in part 1 and part 2 because that really corresponds to my talk here when I talk about the interim data releases that we have for you here today. So the endpoints that we looked at in an earlier data release, it's in blue here. We looked at safety and tolerability, the lowering of HTT protein in Huntington's patients, as well as the exposure of Votoplam in the CNS. Our month 12 endpoints are uniquely different here. So after that data release that we did in June of 2023, we had a lot of questions about, well, what happens after a longer period of time? So we continue to assess safety and tolerability, also looking at the CSF HTT protein lowering along with whole blood HTT protein lowering over a longer spectrum of time as well as other biomarkers. So in addition to what you see here in terms of the initial Pivot HD trial, patients that participate in Pivot HD for the entire year are eligible for another 2 years of long-term extension in which the placebo patients are randomized to either 5 or 10 milligrams. So we offer, in this particular trial, a unique data, or I should say a unique patient population, one that hadn't, at least during the design phase of this program, hadn't really been studied in this way. So we started the trial looking at stage 2 patients according to the HD-ISS, and we used pin stratification in order to try and isolate the most informative population in that stage 2. So I have this next slide here to kind of break that down a little bit because I think it's kind of unique and colorful, if you will. So what we did here is, going back to those days, it was a bit ambitious to go into stage 2 subjects, right? We needed to get clinical information here in phase 2. How could we do this most responsibly? So what we did, and this is really leveraging the work that was done by Jeff Long and Jim Mills, they actually worked with us on this, is we took the entire enrolled data set and we broke it into quintiles, and we said, okay, based on PIN scores, which is the prognostic index that was developed by Jeff Long, which PIN scores would make the most sense to enroll for this trial, which patients that fell into that. So where we found ourselves is we were using the, let's see, pink, blue, and green. And what this graph is showing you here on the left is on the y-axis is the probability of conversion to stage 3. If you remember Jeff's work with the PIN score, originally they went to DCL4. We actually re-engineered that and we're using it to time to conversion to stage 3. So the pink, on a 2-year study, we would expect about 50% of the patients who are participating in the trial to have that conversion event, stage 2 to stage 3. Conversely, in the green line, you're looking at over 4 years before 50% of those patients. And then if we were to go lower in PIN score, those patients would be expected to not convert over to stage 3 for quite some time. So what I'm really trying to say here with this slide is we're very representative of a broad spectrum of HD patients who are clinically symptomatic to be informative for our trial. The last box here that I put there is something that I'll talk about just here briefly. We had originally started the trial just looking at stage 2 patients, and after we got going, we added stage 3 participants via protocol amendment. And as I mentioned before, we stratified by these PIN scores for the stage 2, and we're stratifying by TFC for stage 3. So at the end of the day, our data set will include this broad spectrum of HD patients with different amounts of huntingtin lowering, and I'm going to talk about that right now. So I mentioned before about our week 12 interim readout, and we had specific objectives, which we've met there. I'm not going to read each one of these because this was presented here at HSG last year, but I do want to talk about our objectives for the 12-month interim analysis. So what we really wanted to demonstrate was the durability of huntingtin lowering, all right? We saw it at 3 months. What does it look like at 12 months? We wanted to show additional biomarkers, what the effect was on the biomarkers of interest. And additionally, because we have that additional time, 12 months, we want to take a look at our functional scales and obviously the safety and tolerability for an extended period of time. The enrollees that were evaluated, so I just want to clarify, this is a very small subset of the patient population that are currently enrolled in PIVOT HD. They represent the first 30 subjects that came in. So 10 on placebo, 10 on 5 milligrams, and 12 on 10 milligrams. A bit of a gender disparity at this point, and we don't expect to see that at the end of the trial. Mean CAG length was 44, and the mid-max was 42 to 47. And this is something I kind of want to talk about a little bit too, is originally the protocol, we were only enrolling CAG 42 to 50. And later on, we amended the protocol to include all the way down to 40. However, in this early data set, we don't have those patients represented. Additionally, because we started in the stage 2 population, all of the mean TFC scores are 13, which is, of course, by definition, stage 2. Okay, this graph here that I'm showing, the 12% increase in blood MHTT levels, and then the dose-dependent decrease, 21% and 30%, corresponding to 5 and 10 milligrams, was the data we showed at 3 months, so 12 weeks of treatment. This summer, in June, we added, we took a look at the same cohort of patients after 12 months of treatment. So we're up 14% in placebo, 22%, and 43% on 10 milligrams. So what we believe here, based on the data that we've reviewed so far, is that we were reaching HTT lowering steady state at about 6 to 9 months. So the overall goal for PIVOT HD was to arrive at a dose, or doses, that lowered huntingtin protein between 20 and 50%, and we feel that we've achieved this goal. For those of you who are familiar with our protocol design, originally there was an option to go to a higher arm, so 10 milligrams to 20 milligrams. This was authorized by the DSMB. However, we felt it was in the best interest to evaluate the entire data set before we're pushing the boundaries of huntingtin lowering here by doubling the dose when we've already achieved a mean reduction of 43%. Additionally, with this data release, because we were at 12 months and the way that we wanted to run our CSF MHTT assays, we wanted to run the complete set for the entire year. We were able to release the CSF MHTT lowering here in June. As you can see, we have some really nice concordance in the CSF from what we saw at 12 months with the blood assay. So now turning the page here a little bit away from mechanism of action and going into the safety aspect of this here, I'm showing you on the left the plasma NFL levels at 12 months. So placebo, 5 milligrams and 10 milligrams, all reduced from baseline. But the graph on the right is the CSF NFL trajectories. And in all of our data releases, we only looked at group level data, with the exception of the CSF NFL. We really wanted to understand the individual patient trajectories. Still blinded, what you see is all of the dosing groups there, not differentiated in any way, but as you can see from baseline all the way to month 12, we don't have any increases in CSF NFL and certainly no spikes in CSF NFL readouts at this point with this group of subjects. Looking at the AEs, I don't have too much here to point out other than that the adverse events, if you look by treatment group, were relatively distributed in terms of the treatment emergent adverse events. There is one bit of information, though, that we had not discussed before, but did want to mention here today, is that in the CSF, we've not seen any increases in total protein or any increases in leukocytes. Now, looking at the clinical scales, and I'll kind of go one by one. I think you can see for yourselves what the trends are here, but looking at TMS, we're seeing some nice trends in terms of the slowing of progression on TMS. This is the only the 30 subjects that I mentioned here, but we like what we see here with this interim data release again in 12 months. CUHDRS the same. We had that dose-dependent improvement, or I shouldn't say improvement, dose-dependent delay of worsening, and then TFC. Again, few subjects on TFC, and remember, like I mentioned before, we are in a stage 2 population, so not in that stage 3. Okay, so just really in conclusion, as I mentioned before, we really feel that we achieved all the goals that we set out to do with this interim data release here at 12 months. We look forward here coming up into, oh, I'm sorry, I forgot one thing here. We've gotten a lot of questions here about the United States and PIVOT-HD in the United States. Unfortunately, we had already completed enrollment for PIVOT-HD by the time we were able to have our conversations with the FDA. Those of you who are in the room at sites were with us all along this way, but when we did have this interim data release, of course it went to the FDA and they lifted our partial hold. So that was a really good thing, and I think a lot more optimism, certainly on our part, for trials in the future here in the United States. Furthermore, right after we had that partial hold lifted, we applied for fast track, and fast track was granted a couple weeks after we submitted, or a couple months, I guess it was, after we submitted for that. So this is going to allow us to have much more access to the agency as we have our protocol discussions for phase 3 and interrogate the data from PIVOT-HD and the long-term extension study. So what does that mean here now for PTC and the future of Votoplam? Well, we're going to finish our trial. The data is expected from PIVOT-HD, that year-long trial, in the first half of 2025. And we're not stopping. While patients are still completing this 1 year, we're actively designing and discussing what our phase 3 program might look like. And we hope to get there, contingent, of course, on some great data in 2025 when PIVOT-HD closes down. So in conclusion, again, I want to thank the Huntington Study Group for this opportunity here at the Peter Como Symposium. But mostly, I really want to thank all of our patient participants, caregivers, families, for all of their sacrifices and contributions to our clinical program that got us here to this stage today. Thank you.